Hello everyone, this is Aloysius Guy with you in a new tutorial, and today's tutorial is about a very new immune therapy that is used to treat cancer, and it's called CAR T therapy. So what is CAR T therapy? And as we usually do, we cover what it stands for. So CAR T therapy stands for chimeric antigen receptor T cell therapy. So I know that you have a lot of questions in mind. First of all, maybe the first one is, what does that name mean? Or why T cells are used in that therapy? Why T cells in specific? How does it work? Or why is it effective? Because we know from the news and um, uh, you must have heard about it from all the papers around the world that it is showing a very effective and promising results. So why? Also, is it really flawless? Or does it have some kind of disadvantages and drawbacks? And the most important question is, does it really cure cancer? So we're going to cover all these questions and even more during this tutorial. So before we dive into the process of CAR T therapy and how it works and all these steps of that therapy, we need to understand first, why is it used in treating cancer? And to answer this question, we need to understand something called the tumor microenvironment. So in tumors, you would have a tumor lump. Okay, within that lump, you would have an area, the black one here, which is cancerous, and then you would have a surrounded area which is of healthy tissue. So within this cancerous tissue, you would have cancer cells with immune cells, with fibroblasts, with some extracellular matrix materials like collagen and hyaluronin. Sometimes you would have blood vessels infiltrated within that tumor lump. Okay, so all these things together are called the TME, or the tumor microenvironment. One of the main players within this tumor microenvironment and in tumor progression in general, it's an immune cell called T cell, and they have T cell receptors. And we need to understand the importance of T cells in the tumor microenvironment, and by that, then we can understand their role in CAR T therapy. Okay, so T cells are also called T lymphocytes. So sometimes in other textbooks or maybe in other videos, you might find you might find them calling the T cells T lymphocytes. That's okay. It's just the same thing because T cells are actually lymphocytes, type of lymphocytes. Okay. There are three main type of T cells. So the first one, what we call it cytotoxic or killer T cells, and then the helper T cells, and then regulatory T cells. So the main difference between these three different types is the type of receptors that are on the surface of these cells. So for cytotoxic T cells, they would carry the CD8 receptor. For helper T cells, CD4, and for regulatory T cells, they would have the CD25. Sometimes they will carry also the CD4 or CD8. That's it's a bit different, but mainly they would have the CD25. Okay, carrying different receptors like that would influence the function of every cell type. So cytotoxic T cells, or CD8 positive T cells, they are responsible of killing infected cells or cancerous cells. So for example, if a normal cell turn cancerous, so the T cell will identify it and then interact with it and then kill it. When it comes to T helper cells, their main function is to support the cytotoxic T cells. Also, they are very important in regulating the immune system response, and by secreting specific molecules, they are called cytokines. Okay. For Tregs, their main function is to maintain the immune tolerance for and prevent autoimmunity. So autoimmunity is a different type of disease. It's different from cancer, and it's a disease where the immune cells within the body start attacking the organs within that patient. So it's a bit different to cancer. So now, after knowing the three different types, we need to highlight that the cytotoxic or killer T cells are the ones that are most commonly used in CAR T therapy. Some CAR T therapy use CD4 positive T cells. However, most commonly the cytotoxic T cells are the one used. So now we need to understand why cancer cells induce immune responses. Because as we said earlier, that the T cell is playing a major role in affecting tumor progression. So we need to understand how cancer cells activate these T cells. So at the beginning, what we have is a normal cell. So when the normal cells accumulate a lot of mutations, it turns cancerous, okay? The mutations might occur in so many different genes within the cancer cells. Some genes are called oncogenes and others are called tumor suppressor genes. So when the mutations affect these genes, this results in producing proteins that are in shape a bit different to the normal proteins, okay? And this what it induces the immune response. So if you ask yourself why producing a different shape proteins like different shape receptors or different shape secreted protein might induce an immune response, is because the immune cells, once they interact with these protein or with these receptors, they will identify them as a foreign object, and this will alert and activate the immune response. Okay, that's good. So now we need to understand the role of T cell in that immune response within the tumor microenvironment, okay? So everything starts in what we call a germinal center. So the germinal center, there are places such as the lymph nodes or the spleen, okay? So what happens there is a dendritic cell that has been in a tumor site. It will move from the tumor site and then go to lymph nodes or the spleen, where they interact with T cells that are called naive T cells. So they are inactive T cells, okay? And what happens there is that the dendritic cells will present a cancer-specific antigens to the T cells there. What this does is results in activating the T cells, inducing their proliferation, and then induces their migration to the tumor site. So once the T cells enters the tumor site, they are now called effector cells. So they are activated, okay? And they are usually either CD8 positive or CD4 positive. And once they enter the tumor site, they interact with the tumor cell and then secrete specific factors that kill the cancer cell. And that's why T cells is very important in preventing tumor progression. So the question is, as we said there, So the question is now is, if T cells can interact with cancer cells and kills them, why do we need CAR T therapy in the first place then? Because T cells seems to do the job, right? And we will understand now why is that the case. So although T cells are very good at killing cancer cells and identifying them, cancer cells, because they accumulate mutations, a lot of mutations through time, they will start adapting strategies to evade these cells and prevent T cells from recognizing the cancer cells. And the cancer cells does that through different strategies. And the first strategy is through losing the tumor-specific antigens. So as we said, that each tumor cell would have a specific antigen on its surface that is specific for tumor cells. Once this antigen is identified by dendritic cells will go to the lymph node and then present this antigen to the T cells. So then the T cells enters the tumor site and they will look for cells that carry this antigen. And once they identify these cells, they will kill that cell. So what the cancer cells do is that they stop producing that antigen. So when the T cell come, the T cell cannot identify that antigen. Therefore, they will not identify the cancer cell. Okay. The second strategy is through losing a very important receptor called class 1 MHC receptor. So these receptors are responsible of presenting the antigen. So these antigen, like cancer-specific antigens, these receptors receptors are responsible of presenting them. Okay, so what do the cancer do is that they stop expressing that receptor, and that will result in the same problem, is that the T cell will not be able to identify such a cell. Okay, because the T cell needs to identify that circle antigen to identify the cell as a foreign and then kill it. The third strategy is through inhibiting the T cell and therefore inhibiting the immune response. And this happens through two main ways. So the first way is that the cancer cell will start expressing some inhibitory ligands that will bind to inhibitory receptors on the T cells. And once these inhibitory receptors are activated, the T cell will be inhibited. Okay. The second way is through secreting and releasing a large number of immune suppressive cytokines. Once these cytokines interact with the T cell becomes inactive as well. That's why tumor cells are very strong and very dangerous after accumulating a lot of mutations, because they will start adapting these type of very strong evading strategies. So because of these immune evasion strategies that the cancer cells use, we need CAR T therapy. So now we will cover how CAR T therapy works. But before that, please don't forget to like, comment below, and subscribe, and also to hit the notification button so you can get all the new videos, please. So as we can see here in that image, CAR T therapy, it's mainly split into 5 main steps. Okay, so it starts with taking the T cells from the patient, infecting them with a specific virus, and then creating the CAR T cells, and then expanding them, and then putting them back in the patient. So we will cover each step, and we will understand how each step is very important in that process. Okay, so the very first thing we do, even before we take the T cells out of the patient, is to identify a suitable cancer-specific antigen so that the T cells can target once we put them back in the patient. Okay, so that's a very key step. And these cancer-specific antigens need to have some key characteristics. So as we know that within the cancerous tissue, we would have a lot of cancer cells, okay? And each cancer cell, or a large portion of that cancer cells, will have a cancer-specific antigens. So the first step in that process is to identify a cancer antigen with key characteristics, and these are some key characteristics that we need to look for. So the first thing is that this cancer antigen is constantly expressed on cancer cells. So which means that we need the cells. Most of these cancer cells are always expressing that antigen. Also, it's very preferable to select an antigen that is found in different patients. So the same antigen found in different patients. So by that, we can use the same CAR T cells to help another patient. Okay. We can also, one of the key characteristics is that this antigen needs to be specific to cancer cells, and it has very restricted expression in normal tissue. And the reason for that is that we are trying to avoid CAR T cells from attacking normal cells, because don't forget that the CAR T cells will identify this antigen, and once they identify it, they kill the cell that carry it. So if this antigen is also shared by normal cells, this means the CAR T cell will start attacking normal and healthy cells as well, and we don't need that. The last thing is that we need to make sure that the selected antigen is not prone to loss or downregulation, because as we know now is that one of the main strategies for cancers to evade immune cells is to stop producing that antigen. So we need to choose an antigen that is known to be constantly highly expressed on cancer cells. Okay, And here there are some of the techniques used to identify such an antigen. And this step is very, very crucial to the whole process and to the success for the whole CAR T therapy. Okay, so after determining the antigens that we need to target within the tumor site of that patient, we start with taking the T cells out of the patient in a process called leukapheresis. Okay, so this process involves three main steps. So it starts with drawing the blood from the patient using a machine similar to that one. And what happens is that the blood components will be separated using a centrifuge. And after that, what will happen is that we will be able to collect the white blood cells in one bag and the rest of the blood components like red blood cells, platelets, and plasma in the other bag. And then we will give the rest of the blood components back to the patients, okay? And we will take then the white blood cell and then separate the T cell out of the white blood cell. Okay, so the next step now is to genetically modify the T cells that were collected from the patient and make them start expressing the CAR gene, which is the chimeric antigen receptor gene, and that's done most commonly using viral vector. So the process starts with the collected patient's T cells. We infect them with a viral vector. So in that case, the viral vector will be carrying a CAR gene, okay? And after the virus infects the T cells, the patient's T cells, then the T cells will become CAR T cells, and these cells will be expressing the chimeric antigen receptor that specifically identify the cancer-specific antigen we already chose at the very first step. And that's a very important process. There are other different methods that are used to produce the CAR T cells. So there's non-viral transfection, also CRISPR-Cas9 can be used along with RNA transfection. However, the viral vector transduction is the most commonly used one, and it's very highly effective in producing CAR T cells. So the next step now is CAR T cell infusion into the patient. So after creating the CAR T cells, what happens is that the CAR T cells are expanded and activated in the lab, okay? And then they are infused back into the bloodstream of the patient. However, before the CAR T cells are infused back to the patient, usually cancer patients undertake some type of chemotherapy or radiotherapy, and this will kill a lot of cancer cells and make the tumor microenvironment more favorable once the CAR T arrives. So they will be able, so the CAR T cells will be able to divide and also exert its anti-immune effect within the tumor microenvironment. So now we will cover how the CAR T cells target cancer cells. So after infusing the CAR T cells into the bloodstream, the CAR T cells will start circulating the bloodstream of the cancer patient along with blood cells and other white blood cells, okay? And as this CAR T cells circulating the body, they will come by the tumor site. And once they are by the tumor site, CAR T cells will move out of the blood vessel, enter the tumor site, and once they are there, they are carrying the chimeric antigen receptor that is specific to cancer cell. Therefore, they will specifically bind to cancer cells and kill them. And that will be the final step that hopefully will cure the patient from cancer, or at least kill the vast majority of cancer cells. Okay, so after all, CAR T therapy isn't flawless. It has some advantages and disadvantages. So the main advantages of CAR T therapy is it has a high response rate, it's potentially curative, it's personalized treatment, which means it cures a specific cancer that that patient has. It's a single infusion, so it's just a single time you put the CAR T cells into the bloodstream and that's it. Also, it has a minimal cross-resistance, which means there isn't a high chance that the cancer will become resistant to this CAR-T cells. However, some of the disadvantages are it's very expensive. It has a very limited effect on solid tumors. So currently, it's mainly used in treatments of leukemia, but it still needs some improvement with solid tumors. Also, it might induce something called cytokine release syndrome, which is like an immune reaction that in some cases might be life-threatening. And sometimes it might result in off-tumor toxicity, which means that the CAR-T cells might identify normal healthy cells as foreign and start killing them as well. So these are the main and the key advantages and disadvantages of this new therapy. So despite all these disadvantages, this therapy holds a very promising future and showed very promising results as well. So in 2022, The Guardian has published that two of the very first human being who suffered from leukemia, and they were treated with CAR-T therapy back in 2010, they were still, until the day of that news was published, they were still healthy, and there was no sign of returning of their leukemia, so they are potentially cured from cancer. That's why the world currently is putting a lot of money, a lot of efforts in improving this therapy, and hopefully they might make it, hopefully in the future, a bit cheaper, because it holds a lot of promising results. And as always, here are some questions that will test your understanding of today's tutorial. And now we came to the end of this tutorial. I hope you enjoyed it, guys, and please don't forget to like, comment, subscribe, and also to hit the notification button so you will be notified of our upcoming future videos. Thanks for watching.